Okay, so great to be here. Thank you very much to the organisers for the invitation. I'm really excited to be here. So I'm going to talk about the work that we've been doing for the last few years in Manchester and really understanding mechanisms of inflammation that contribute to brain injury and understanding primarily inflammasomes. So I think of inflammation as a self-amplifying loop. This is on the left of the slide here. This is a rat brain that's had a stroke. Now, it's been stained with a vital dye, and the pale area is an area of necrosis and cell death. And what happens when you have areas of necrosis is you get the release of danger signals. These are normally intracellular molecules that are not sensed by the immune system, and when they come out following cell death, the immune system recognizes that something drastic has happened and responds. And the response is the sensing of PAMPs, or if it was an infection, pathogen-associated molecular patterns, or PAMPs, they're sensing by pattern recognition receptors on cells of the innate immune system, and in the brain, these are, of course, microglia. PAMP and DAMP sensing results in a cascade of signaling events that results in the production of inflammatory cytokines, such as members of the interleukin-1 family, and this then triggers the recruitment of inflammatory cells. And they come in geared with processes to kill pathogens or pathogen-infected cells. Of course, in a stroke or a non-communicable disease, there isn't a pathogen to kill. You just get collateral tissue damage, and the cycle continues. And one of the major DAMP sensing pathways is the NLRP3 inflammasome. So NLRP3 is a pattern recognition receptor or sensor, and it's implicated in a lot of non-communicable disease states. If you look at the left-hand side of the slide, this really illustrates the canonical pathway of NLRP3 activation. So there's a couple of other pathways. I'm only going to talk really about the canonical pathway. And it occurs in two steps. The first step is a priming step, and this is where the cell, the microglia or the macrophage, senses the PAMP or the DAMP, typically through TLR signaling, and you get the induction of pro-inflammatory gene expression. This is typically NLRP3 itself, and members of the interleukin-1 family, interleukin-1 beta and interleukin-18, which are expressed as inactive precursors. And this primed cell then requires to encounter a second stimulus. And there are a number of different stimuli that can activate NLRP3. Now, none are described to directly interact with NLRP3. Rather, they converge on some signal that results in the activation, the post-translational modification of NLRP3 and its activation. And in the inflammasome field, it's been quite an open question how NLRP3 senses these diverse signals, and I'll talk a little bit about that. Once activated, NLRP3 binds to an adapter protein called ASC and nucleates its oligomerization. And this forms a large scaffold upon which the protease caspase-1 is recruited and activated. An active caspase-1 will then cleave the cytokine precursors to their active forms. It also cleaves a protein called gasdermin D, the N-terminus of which will oligomerize, embed in membranes, and form pores, which serve as the conduit for interleukin-1 secretion, and also leads to the pyroptotic cell death. This is a rapid pro-inflammatory form of cell death. And once extracellular, interleukin-1 can then exert its inflammatory actions. On the right, just to give you an idea of scale, this is a microglial cell from an organotypic hippocampal slice culture where we've activated inflammasomes, and you can see this speck here. That's an inflammasome complex. So they're enormous complexes with respect to the size of the cell. So as I mentioned, one of the questions that's been in the field is how do these diverse stimuli activate NLRP3? And we've been very interested in this because we think if we can understand NLRP3, we can target it in models of brain inflammation, which I'll illustrate in a few minutes. A few years ago, James Chen published this brilliant paper about the mechanism of NLRP3 activation, which suggested that it was the fragmentation of the Golgi apparatus that was important. We work quite closely with cell biologists in Manchester, and we thought this possibly wasn't the case. A lot of the TGM fragmentation work has depended upon TGM46, which is in the Golgi, but it cycles to the plasma membrane and back to the TGN via endosomes. And there's a lot of evidence that whenever there's a disruption, the trafficking of endosomal cargoes, the TGM46 gets trapped in endosomes, and that gives it this vesicular appearance. So this was the hypothesis we wanted to test. This work was published earlier this year. Dan and Bali led most of the early work here, and others have contributed massively to this. And the way we studied this initially was we looked at TGM46 in the cell. Remember, this cycles via the endosomes to the plasma membrane and back. And we looked at Golgi-97. Golgi-97 is a permanently resident TGM protein, and we looked at their co-localization. When we activated the inflammasome, here we've used nigericin, we get this dispersal of the TGM46, this vesiculation, but the Golgi-97 signal remains intact. And when we look at the co-localization analysis, we see this. We see a drop in the co-localization, so these two proteins are separating, and which is consistent with, not with the TGM fragmentation, but with the TGM46 getting trapped in endosomes. Conversely, when we look at the co-localization analysis with endosomal markers, here we're looking at EEA1, which is a marker of early endosomes, we see an increase in co-localization, suggesting that when we're activating the cell with inflammasome activators, we are getting a disruption in the trafficking of endosomal cargoes. When we looked at NLRP3 in these cells, this is NLRP3 labeled with M venus, which is a fluorescent protein. At rest, the NLRP3 is largely cytosolic, but you do see a localization on the TGN. When we activate the cells, we get the TGN dispersal, TGM46 signal dispersal as described, and we see a co-localization now between the TGN and the NLRP3 with markers of the endolysosomal system. So endosomes, I'm showing here CD63, which is the marker of late endosomes, and also LAMP1, which is the marker of lysosomes. So when we disrupt the trafficking of endosomal cargoes, we see the activation of we see NLRP3 decorating vesicles of the endolysosomal system. There has been work, we've done some work, and others have done some work, and James Chen initially described the importance of the lipid PI4P involved in NLRP3 recruitment to membranes. We think that's the case, but we also think other things could be involved. So this is preliminary work. We haven't published this. This is work by Rose Wellens, who's just finishing her PhD in my lab. And Rose, to attack this, Rose has been doing some proximity labeling. So Rose has expressed NLRP3 bound to a protein called turbo-ID, which is a biotin ligase. So that means when we add biotin to the cell, this enzyme will biotinylate any proteins that interact with NLRP3 or are in close proximity. So we looked at conditions of rest and NLRP3 activation. We added biotin. We then did a streptavidin pull down and analyzed the hits by mass spectrometry, and we found a number of proteins that were significantly enriched that are involved in cargo trafficking inside cells. So as I said, this is preliminary, but we've done some work to study this further. One of these hits that was really interesting for us was this protein TPD54, also known as TPD52L2. Now, there's virtually nothing known, there's very little known in the literature, but what is known has been done mainly by Steve Royal at Warwick, who's published this paper, using TPD54 as the defining characteristic of intracellular nanovesicles. And these are tiny vesicles that shuttle between organelles in the cell, trafficking cargoes. And what was really interesting for us was that when Steve knocked down when he knocked down TPD54, he dispersed the TGM46 signal, which is consistent with the phenotype we see when we activate the inflammasome. So we've done this very recently. We used CRISPR to knock out TPD52L2 in THP1 monocytes, and we see an activated inflammasome response. So we spend a lot of time looking at the cell biology and the biochemistry of inflammasome activation. We think that its activation is dependent upon the sensing of a disturbed cellular homeostasis, which is manifest by a disruption of intracellular cargo trafficking. And we think that the endolysosomes serve as a platform for the activation of NLRP3. And our proximity labeling is identifying potentially new interactors that we can identify as therapeutic targets. We think this is important. We think we need to study this because NLRP3 is reported to contribute to a number of brain diseases, or the inflammation in brain diseases. And I'm going to give some examples where that's been the case. This work I'm going to describe was based on the work of Michael Henniker. I don't know if Michael's here yet, but it was terrific work in the field, and it established that you've got active inflammasomes in the brains of people with Alzheimer's disease. And in the APP-PS1 mouse, the memory deficit was dependent upon the NLRP3 inflammasome. And we'd been interested in this in a number of ways, and one of the ways was work we'd previously published showed that zinc depletion in macrophages and microglia, or zinc deficiency, was a stimulus for NLRP3 inflammasome activation. And we think zinc deficiency could be quite a common comorbidity in people with Alzheimer's disease. And there's certainly some reports in the literature suggesting that people with Alzheimer's disease are zinc deficient, and sometimes aged people can be zinc deficient. So we wondered whether zinc deficiency could accelerate the memory decline in Alzheimer's disease, and whether that's something that could be readily fixed through a dietary intervention. So this was the experiment we did. We published this a couple of years ago, actually. This was work by Jack Rivasodi, who was a postdoc in the labs of myself and Catherine Lawrence. Jack's now a lecturer in Tasmania. And the experiment Jack did was he took control mice and he took APP-PS1 mice, and he put them on, at 3 months of age, he put them on a control diet, which had 30 mg per kg of zinc, or he put them on a zinc-deficient diet, which had 3 mg per kg of zinc. And he put them on that diet for 6 months. In one group, he put them on the zinc-deficient diet for 3 months and then switched them back to the normal zinc diet. And he did a battery of memory tests every 3 months. This is the Y maze he's done here. And in the control mice, there was no effect of diet on memory, whether it was the normal zinc diet or the zinc-deficient diet. On the APP-PS1 mice, on the control diet, there was no effect on memory. And we expected that because these mice don't really develop deficits until 12 months plus. So at 9 months of age, there's no deficit. However, on the zinc-deficient diet, after 3 months on the diet, they do have a deficit, and that's further pronounced at 6 months on the diet. And this was reversible. So when we put them on the zinc-deficient diet for 3 months and then put them back on the normal zinc diet, the memory was recovered, suggesting that the zinc deficiency is accelerating a cognitive impairment or a memory impairment in these animals. So we asked the question, is this NLRP3 dependent? So the experiment, we repeated this experiment, but basically we crossed these mice with mice that were knocked out for NLRP3. And these are the data. If you look at panel A here, on the normal zinc diet, whether there's NLRP3 present there or not, of course, there's no deficit consistent with what we've seen before. On the zinc-deficient diet, in mice that were wild-type for NLRP3, there is a memory deficit. And this is abolished when NLRP3 is knocked out. So NLRP3-dependent inflammation, or an NLRP3-dependent process, is driving this acceleration of the memory deficit. Now, we don't quite know how this is happening. We looked at microglial activation crudely compared to the methods described yesterday. We didn't see any difference, and we didn't see a difference in plaque burden. So we don't really understand it yet, but this is an area that we and many others are actively studying. I'm going to talk about a different condition now where we know that NLRP3 is important, just to highlight some of the other work that we're doing. And this is work I've been doing with Kevin Cooper, who's an immunologist in Manchester. And Kevin's he works on cerebral malaria, which is responsible for the deaths of hundreds of thousands of children in Africa every year. And we can model this condition by infecting mice with red blood cells infected with this parasite, Plasmodium berghei anca. And this fairly well recapitulates many of the aspects of the human cerebral malaria condition. So we get an increase in parasitemia, and the mice fairly quickly succumb to this and die, and they die by brain swelling and brainstem herniation. The way this is treated with antimalarial drugs, and we can do this in the mice as well. These are mice that have been treated with artesunate and chloroquine antimalarial drugs. These kill the parasite, and they reduce the mortality of the syndrome. Not completely, but they reduce it and they recover the behavior. Now, we knew that NLRP3 is not important for the development of this syndrome. But what we'd found was that when we gave the antimalarial drugs, and these are the brains of mice that we infected, these were Ask citrine mice, so these were Ask reporter mice, where we could monitor specks in vivo by fluorescence, the mice that Doug Golenbach developed. We did see specks that we think are in microglia, monocytes in the vasculature. We subsequently found that the antimalarial drugs could activate the inflammasomes in cells in culture. And when we gave the NLRP3 inhibitor, MCC950, as an adjunct therapy with the antimalarial drug, we improved the survival of the treatment further, suggesting that the antimalarial drug treatment could be having an iatrogenic effect. And so the efficacy of this treatment could be reduced by its effect on inflammasome activation. So we're working on this just now. We don't understand this fully, but we're starting to get to the bottom of it. So in this experiment, when we give the animals the antimalarial drug, when they start to get sick, we then give them, we inject them IV with FITC, and we can see the permeability, how much water is getting into the brain, and you can see pretty clearly the blood-brain barrier is really massively disrupted in these animals. And it recovers quickly. It's quite remarkable. So this is the FITC fluorescence. It correlates with brain water content. So we get this swelling, and when we inhibit the inflammasome, we see much less permeability of the blood-brain barrier. So we think that the inflammasome, potentially via an interleukin-1 dependent process, is acting at the blood-brain barrier to drive this effect that we observe. So clearly understanding the NLRP3 inflammasome is important, because if we can identify ways of targeting it, we can find ways of potentially treating disease better. So developing NLRP3 inhibitors is a big area. There's a lot of people interested in it. I'm just going to highlight a couple of things that we've done. The drug I've described, MCC950, that was originally a Pfizer drug. That's the kind of gold standard tool. Many people have used it. It's been further developed by, recharacterized really by Luke O'Neill and his colleagues. Some of our own efforts in this area, we do a fair bit of chemical biology in Manchester, and these are some of our drugs. This MBC series are direct inhibitors of NLRP3, recharacterized. And through understanding an importance of chloride channels in regulating the canonical NLRP3 inflammasome, we developed our MVR series, which doesn't directly inhibit NLRP3, it only targets the canonical pathway, which could be useful in some contexts. So just to summarize this part of the talk, NLRP3 dependent inflammation contributes in a number of models of brain disease, and targeting it could be really beneficial. And this is an area that we and others are very interested in. Another condition that we've done a lot of work on in Manchester is stroke, ischemic stroke. And that, we know that is interleukin-1 dependent. If we give anakinra in an animal model, the animals are really protected. We get a big reduction in the infarct. And we've currently, my colleagues in Manchester have been running clinical trials in stroke and hemorrhagic stroke with anakinra for many years. And they've just completed a phase 3 trial that they're waiting to read out. So we have access to a lot of stroke models and expertise, and we've been studying this. This is work by Eloise Lemarchand, who was a postdoc in my lab. She's now a fellow in Cannes. And knowing that interleukin-1 was important, we hypothesized inflammasomes are going to be important, and that surely NLRP3 would be important. This is the model of stroke that Eloise used. She exposed a bit of the brain. She dropped ferric chloride onto the artery. This caused platelet aggregation and the formation of a clot, and you get an infarct. So this was the experiment Eloise did. She gave these animals a stroke and then used MCC950, the NLRP3 inhibitor. She's given it in this experiment IP. She also reproduced the same experiment by giving it ICV, and there was no difference in infarct volume. We looked at wild type and NLRP3 knockout mice, littermate controlled, and there was no difference, and we gave them a stroke, and there was no difference in infarct volume. So at least the primary injury in ischemic stroke doesn't appear to be NLRP3 dependent. Now, we know a lot of the conditions that predispose people to strokes or can increase stroke injury could be NLRP3 dependent, at least the primary injury in these control mice doesn't appear to be. But it led us to then question, well, what inflammasome could be important? So NLRP3 is one pattern recognition receptor that can form inflammasomes. There are others. And for the last part of my talk, I just want to briefly talk about some recent work we've done on a different one called AIM2. This is absent in melanoma 2. This is a DNA-sensing inflammasome, so it has two domains, a Hin domain that binds to the double-stranded DNA, which can come from pathogens or it can come from host cells that have been ruptured. And it has a Pirin domain that then binds to the same adaptor protein NLRP3 does, which is ASC, and it will then oligomerize, form inflammasomes, where you get interleukin-1 processing and pyroptotic cell death. And if this graph at the bottom here, when we compared, we used a different model of stroke, this time a filament occlusion. When we looked at wild-type and AIM2 knockout, we got a big protection when we knocked out the AIM2 inflammasome here. We can activate AIM2 in culture. We just transfect the cells with poly-DADT, and we get a pretty good AIM2 response. And from a screen we'd been doing, we identified this molecule, 4-sulfonic-calix-6-arene, as an AIM2 inflammasome inhibitor. And you can see that this molecule pretty well blocked the AIM2-dependent pyroptosis and interleukin-1 beta release. There's a number of ways you can look. You can measure inflammasome activation. You can look at specs via microscopy. If you look at the gels here on the right, you can see at the top here we can measure ASC oligomers in the gel and interleukin-1 processing, gasdermin processing. So when we transfect the poly-DADT, we get the same AIM2-dependent effect. The calixarene blocks this, and the NLRP3 inhibitor has no effect, so it's quite a specific response. We wondered how this molecule might be working. We made this reasonable assumption that the negative sulfonic groups of the calixarene could mimic the negative phosphate groups of the DNA, and it could be disrupting the DNA binding site of the Hin domain. To test this, we did the following experiment. Thinking that charge was important, we took a 4-sulfonic-calix-4-arene, which has two less negative charges, and we took the 4-sulfonic-calix-8-arene, which has two more negative charges. And then we also had an uncharged compound, the 4-tertiary butyl molecule, which has no charge. So we could really test this hypothesis. We were able to model this computationally, and the binding energy suggested certainly that charge was going to be important to the DNA binding site. And when we did the experiment, this is AIM2 activation with the readout interleukin-1 beta release. You can see that the uncharged molecule had no effect whatsoever. And as we increased the charge, we increased the potency of the molecule. And we've got, in this cellular assay, we've got an IC50 of 210 nanomolar for an AIM2 inflammasome inhibitor, which is about the best out there currently. Just to confirm that this was interacting with the DNA binding site, we did this simple gel shift assay. When we run the DNA through this gel, you can see it migrates. In the presence of recombinant AIM2 protein, that migration is inhibited. But then when we incubate with these different calixarenes, we displace the DNA from the AIM2. And you can see this is correlating with the increase in potency. So it's a DNA that's binding to the Hin domain of the AIM2 to inhibit it. We wanted to test this in a disease model. To do this, we worked with Stefan Roth and Arthur Leitz, and a couple of years ago, they published this really interesting study. And it's known that after a stroke, after a brain injury, you have a period of immunosuppression, and this is a big problem because it can result in post-stroke infections and complications. And this, Arthur discovered, is dependent upon the AIM2 inflammasome. There's this, after the injury, there's an increase in cell-free DNA, and this gets taken up by myeloid cells. You get AIM2 inflammasome activation, and ultimately resulting in a fast ligand-dependent T-cell death and this immunosuppression. And we used this model. We worked with Arthur. We gave the animals our AIM2 inflammasome inhibitor, and we rescued T-cell survival in this model, preventing immunosuppression. And we've done a few other studies around that as well, suggesting that targeting AIM2 could be therapeutic in a number of contexts. So just to summarize this, we think that stroke, the acute injury is not NLRP3 dependent, but there's a number of outcomes of stroke that are dependent on inflammation, and potentially we think the AIM2 inflammasome is important here. And developing inhibitors for NLRP3 and AIM2 dependent inflammation could have a number of therapeutic applications, and that's where our interest lies here. So just to finally thank everyone who did the work. It's a big team effort. Thank you to my collaborators and the funders, and thank you all for listening.
Thank you for presentation, and please, questions?
Great talk, David.
Thank you.
When you get inflammasome activation, do you always see both IL-1 beta and IL-18 secretion?
Not always.
Do you get one?
There's some conditions, so I haven't shown, but we have a model of hyperinflammation in the lab, which is a repeated CPG injection, and actually that appears to be predominantly IL-18. We don't pick up any interleukin-1 in that, but it's the IL-18's inflammasome dependent. So I think there are contexts where you get an IL-18 specific response. And IL-18 is constitutively present. It doesn't need to be induced by the priming step like interleukin-1 beta. So,
Because we find we cannot measure IL and 18 secretion in our microglia
right.
when you get inflammasome activation. We only get IL.
Okay. Well, that's interesting.
Yeah.
Yeah. Okay, I'll have a think about that. Yeah.
Thank you. Do you have any idea if zinc impacts on cargo and intracellular organelles?
Yeah. Zinc, it's actually stored in lysosomes, and so we imagine that it potentially depleting cells of zinc will disrupt cargo trafficking, but it's an experiment we haven't done yet, but we will do it. Yeah. Thank you.
So, thank you so much for the talk. And we also know that potassium efflux is necessary for the inflammasome activation. So is it possible that zinc and potassium both are positively charged? So if you supplement the diet with potassium, do we see the same effect? Is it the cells trying to compensate for the extracellular potassium by zinc? Is it possible?
I don't think so, just because the levels are so vastly different. You know, the levels that change for potassium to activate the inflammasome, we're talking orders of magnitude different. So I don't think that's how zinc's working.
Okay, thank you.
Thank you very much.
Thank you.